Hey, Stefan, why aren't you here to do this intro? Okay, like 3 months ago, Stefan told me that you need to invite somebody doing quantum biology because I'm supposedly a quantum girl here. I was told, right? So I got, live things and me that doesn't go together. I don't like to study that. But anyway, so jokes aside, it's really a great pleasure for having Clarice Aiello. Am I pronouncing your last name correctly? Yeah. Here. She's coming to us from, well, we taught UCLA, but now from Quantum Tech. This company who's basically, as you're going to hear, using quantum effects and spin effects to both understand and then, make useful things by mimicking biological system. And, the funny thing is that I just found out when I had to do this intro and prepare and look at her CV that, of course, Clarice is, she did her undergraduate studies at École Polytechnique. For those of you who speak French or know this, we bow to these people who finished Polytechnicien. And then, of course, after finishing École Polytechnique and physics there, she was, where else do you go to do your PhD but to MIT? And she was a student of David Corey, but then he left MIT for quantum QIC in Waterloo, or his empire there. And I just found out that Clarice was a first graduate student of Paola Capellaro. If you know, we just had her like a month ago, or 2-3 weeks ago. And Paola was talking to us about this quantum sensing, how to use that and to measure magnetic fields. And there it is, what happens, with your students afterwards. They use magnetic fields to manipulate or understand biological objects. Or as we always say, we want to copy the nature. And nature is quantum mechanical and the most efficient machine. So we just need to learn how to copy it. But so without further ado, I'm going to stop talking now about this, and I let Clarice tell us all about this amazing work and in her company now, right?
Yes.
So here.
Thank you all so much for being here. I am trained as a physicist, and I really appreciate talking to physicists because what I'm going to talk to you about is a overlooked, understudied, and very often misunderstood degree of freedom in biology that seems to be quantum in nature. If you will, to an audience of physicists, I would describe it as light-matter interactions where matter is biomatter, okay? And it turns out that what I'm going to talk about is the fact that there is no doubt that biology and all the machinery of the cell that you can see depicted in my slide, all of these biological phenomena seem to depend on weak magnetic fields in a way that is consistent with some quantumness driving this phenomenon. The machinery of the cell responds to weak magnetic fields in a way that I'm going to explain more about. I'm also going to tell you a little bit about what data in this field looks like and how I think we can break clean with this old way of understanding and looking at this phenomenon by building what I'm calling quantum instrumentation to give us the code book on how to deterministically tweak these degrees of freedom in biology that seem to be quantum. And finally, it's really important that I transmit the message that this is not a basic science curiosity. So I really do think that within 1 or 2 decades, we might be able to harness what I'm calling endogenous. There's no need for genetic engineering. Non-chemical, everything is electromagnetic. Non-invasive, no need for surgery. Cheap, remotely actuated electromagnetic interventions that can inform a myriad of fields, from therapeutics to space travel. And yes, we're going to talk about space travel. It's going to be quite a ride. Okay, but first of all, I would like to thank the people who have been working with me on that. They're the real heroes of this story. Okay, so I like to call myself a quantum engineer. This means that I build instruments to study and control things that are so small and so well protected from their environment that they're better described by the laws of quantum mechanics, as opposed to the laws of classical mechanics that rule everything big around us. So people get a little bit surprised when I say that I study things that possibly happen inside birds and, in fact, inside all our cells. And that's because I'm a quantum engineer who's interested in how quantum physics informs biology at the nanoscale. So before I explain where I'm going, I really need to tell you where I come from. I am trained in this field called quantum sensing. You can mathematically prove that if you use a quantum object, such as an electron, as a sensor, your measurement is improved. In other words, the sensor's quantumness enhances the measurement. In my past, it's very good that Paula was here 2 weeks ago because it gives the context to my talk. I was trained in a type of technological quantum sensor that works at room temperature and under messy conditions. In the messy conditions of the diamond sample within which it lives, or in the messy conditions of samples that it wants to probe. So I am trained in the field of quantum sensing with electron spins in diamond. But my talk today starts in the middle picture there. In fact, I'm going to show you that there is unambiguous evidence that electron spins inside proteins are behaving as true bona fide quantum sensors. I'm going to spend a lot of time in there, so you can expect data to only start showing up 25 minutes from now, because I really want you to know the background of this field. But I'm going to talk about, by the end, on the correlative, incomplete evidence that it seems that electron spins are also working as quantum sensors inside proteins, inside cells. So bear with me. The introduction is going to be a little bit long, but there's a reason why I am doing this. Okay, so let's start with the chemistry. So it's been known by chemists for more than 40 years that weak magnetic fields can alter the final products of a broad class of chemical reactions that depend on electron spins. Here's how it happens chemically. There's a chemical reaction that is happening, it's doing its thing, and at some point the chemical reaction comes to a crossroads. At that point, the chemical reaction is actually going to look for the spin state of a particular electron. If the spin state of the particular electron is measured by the environment to be up, to be down, the chemical reaction continues through one branch, giving rise to the products marked in yellow. However, if the electron spin that the chemical reaction cares about is measured by the environment to be up, the chemical reaction continues through a completely different pathway, giving rise to the red products. But, as you know, electrons are quantum beasts, which means that in the presence of a magnetic field, a spin-down electron can be put into a superposition state of both up and down. Okay? So sometimes I give this talk to a non-physics audience, then the analogy that I use is that a superposition state can be understood as like the electron being in a dimmer switch sort of position where it's both up and down at the same time. And after some time, of course, everything that starts quantum dies classical. The electron is better described again by the laws of classical mechanics, a flip switch. It's either up or down. But for a short time, a magnetic field will put the spin into a superposition state. And the idea is that if there is one molecule undergoing this chemical reaction many, many times, or many molecules undergoing this chemical reaction once, on average, the final products of a chemical reaction in the presence of magnetic field blue is going to be given by a combination of products of only spin up and a combination of products of only spin down by a proportion that has to do with the superposition state, the position of the dimer, which is to say that another different green magnetic field will yield slightly different combinations of final products. It's in this way that magnetic fields can be actually alter the macroscopic final products of this type of chemical reaction. Now, all of this is absolutely true. There are chemical reactions that depend on that. But when we talk about chemical reactions of this type that are important for biological function, this is the picture. The chemical reaction usually cares about the spin state of two electrons. The spin states can either be in a singlet or in a triplet state. Now, this is a bit of a simplification. By the singlet state, it's the usual singlet state. It's up, down, minus, down, up. And the triplet state that I put there is the triplet 0. It's down, up, plus, up, down. Okay? But everything else is the same. It's just that the chemical reaction cares about 2 spins. Some observations that are super relevant. Even though the interaction with the magnetic field is super brief, the consequences of this interaction are felt at much longer time scales after the spins are, again, better described as a flip switch, okay? Because everything happens at a crossroads in this chemical reaction. So the macroscopic consequences are felt downstream after coherence is lost. The second point, which is really the most technical point that I'm going to make, and probably the most beautiful too. This type of phenomena is markedly different from electron paramagnetic resonance and nuclear magnetic resonance, in particular for a reason that is counterintuitive and that I'm going to explain in a second. The magnetic fields that can actually alter the final products of this type of chemical reaction are relatively weak, on the order of millitesla, on the order of hyperfine interactions. That is, the magnetic field that nuclear spins produce and affect electron spins. And here's why, okay? This is super counterintuitive. So if you're starting to doze off, wake up again. Okay, here's what I mean by that. I mean that if you have 2 very big magnetic fields, say big magnetic field blue and big magnetic field green, the final products are going to be similar. Only tiny magnetic fields can actually alter the final products. And this whole thing only works because and while the 2 spins that the chemical reaction care about see slightly different local magnetic field environments. Okay? In particular, the electrons involved in the reaction that the reaction care about might see nearby nuclear spins that produce a hyperfine field. In this case, the leftmost spin is very weakly hyperfine coupled. It sees only a couple of nearby nuclear spins, whereas the rightmost electron spin sees a lot of nuclear spins. Okay? And as you know, in the presence of a magnetic field, say, in the vertical direction, an electron spin will Larmor precess around it with a frequency that will depend on the strength of the field. The larger the field, the faster this precession, right? So in this picture, the leftmost spin is precessing slowly, whereas the rightmost spin is precessing faster. Now, just to make things simpler, let's assume that the leftmost spin is not hyperfine coupled at all. Only the rightmost spin sees an internal magnetic field produced by nearby nuclear spins. If we add a tiny external magnetic field, the blue magnetic field, well, the leftmost spin is going to start Larmor precessing with the frequency given by this external field, because that's the only one that it sees, whereas the rightmost spin is going to Larmor precess with a frequency that is a combination of the hyperfine field and this external magnetic field. Now let's go to some extreme here. Let's assume, first of all, that this external field is very, very tiny. In particular, it's way tinier than the hyperfine field seen by the rightmost spin. In this case, the leftmost spin is going to start Larmor precessing because it sees this tiny field and it makes a difference, whereas the rightmost spin almost does not care about this tiny field because it's so much more strongly magnetically coupled to the nearby nuclear spin. So its precession frequency will almost not change. Okay? Now, if we wobble, if we change a little bit this tiny field, well, the leftmost spin is going to sense this difference, to quantum sense this difference in this tiny electromagnetic field, whereas the rightmost spin will keep on not caring about it. So by changing a little bit this tiny magnetic field, there is a pickup of a phase difference in the precession of those fields. In fact, open parenthesis, if you remember from your quantum class, Ramsey interferometry. In Ramsey interferometry, you take a spin, you put it in a superposition of up and down, and you let it evolve under the presence of a magnetic field. Component up evolves differently in the presence of magnetic field than component down, which gives rise to a relative phase between up and down in a way that depends on this field. This is actually the two-spin generalization of Ramsey interferometry, close parentheses. Here's what this means. This means that a tiny field puts the spins into a superposition state of triplet and singlet. A little change in this tiny magnetic field, because of the quantum sensing work that the leftmost spin is doing, is going to impose a relative phase between those two singlet and triplet states by a phase factor that depends on this tiny change in magnetic field. If you remember, differences in phase are experimentally mapped into different probabilities that the spins are found as triplet or singlet in a way that depends on this tiny magnetic field. So if you plot the expected final products of the chemical reaction, they're going to vary between the products of only spin up, or only spin singlet, spin triplet, and only spin triplet, singlet, by a proportion that actually is a function of this magnetic field. Okay, so if you will, it's like a two-spin Ramsey interferometry to some extent. Now, what happens if we have a big magnetic field that in fact is way larger than the hyperfine interaction? In this case, both spins are going to start Larmor processing with a frequency highly influenced by this big external magnetic field. So that if you wobble a little bit this big magnetic field, both spins are going to react in the same way, meaning there is not going to be a relative phase difference picked up by those two spins. In other words, big magnetic field puts the spins in a superposition of singlet and triplet. You wobble it a little bit, there is no phase picked up between those two states, so that the probability that the electrons are found in a triplet or in a singlet is not going to depend on this tiny variation of magnetic field, and in fact is going to be fixed by this large magnetic field. So that, and that's very important, so that it gets its own exclamation mark, you get a very interesting, super counterintuitive picture whereby you have an effect magnitude or a magnetosensitivity as a function of external magnetic field that does not only go up, does not only go down, it goes up and down. So in particular, for magnetic fields that are way larger than the internal hyperfine interaction, the magnitude of the effect goes to 0. Okay? So parenthesis, asterisk, if you keep on going, at the level of, say, a couple of Tesla, this curve may go up again due to another mechanism having to do with the fact that the two electrons have slightly different G factors. Close parenthesis, this is just for the experts, but for all intents and purposes, weak magnetic fields are what are changing, are the ones that have the capability of changing the equilibria of these chemical reactions. This is super, super cool. So finally, and that's extremely important, and I've never seen it put it quite this way. Well, in its simplest form, quantum sensing is actually using superpositions as a resource to do the sensing. There are more complicated quantum sensing schemes that depend on entanglement, that depend on squeezing, but in its simplest form, a quantum physicist would call that quantum sensing. Chemists don't call it like this, but again, this is bona fide quantum sensing. This is superpositions being used as a resource. Once the superpositions die out, that ceases to matter for the outcome of the chemical reaction. So again, bona fide quantum sensing. Okay? So this has been unambiguously shown in the solid state, in the gas phase, in the liquid phase, and for magnetic fields as tiny as the Earth's, which again is orders of magnitude smaller than your cell phones. No doubt that this happens at room temperature for things, and especially for biocompounds in solution. Now, I really don't care about birds, and you're going to see how much I really don't care about birds. But this whole thing goes from the chemistry to biology because of birds. I'm only here because of birds. Because birds have an internal compass. There's no doubt, again, that when birds migrate north-south, they do so using, at least as a partial cue, the tiny magnetic field of the Earth. Okay? And people have been trying, again, for decades, to understand how they do so. And none of the theories actually withstood experiments with birds. Now, in 1978 came this brave, crazy theoretical biophysicist named Klaus Schulten. Klaus Schulten made an outrageous hypothesis. Here's what he said. Well, were the same type of electron spin-dependent chemical reaction be happening inside cells, inside birds, birds and other organisms could maybe sense magnetic fields to the extent that they could sense different physiological concentrations of products coming from this type of chemical reaction. So maybe a bird turning right, it would see certain magnetic field lines. Turning left, it would see some other magnetic field lines. And maybe, okay, just hypothesis, in their eye there would be something that would photomodulate like how bright they see, so that they would know they would need to follow, say, the brightest image. For birds, nobody knows if that's what's happening, because it's very hard to go from the chemistry to the behavior, okay, of an animal. But I'm going to show you that actually this is a theory that is withstanding the trial of time. However, this theory is downright outrageous. And sometimes when I give this talk to a physics audience, there's already like 5 people angry at me, shouting insults at me, like, what are you doing? Okay, so here's why. So again, the profound implication that, again, I have never seen it put it quite this way. If birds are truly using this mechanism to fly, to orient themselves, the profound implication is that electron spin superpositions are maintained inside cells, inside birds, for long enough for it to matter for function, in particular to help birds orient. Let me tell you what I mean by long enough. You all know that after some time, quantumness is lost, right? So again, I spent my whole PhD doing better measures on how to do this. But back of the envelope calculation, you can say that a free electron can sense a magnetic field if it can learn or precess once before undergoing decoherence. Well, for the magnetic field of the Earth, which is 50 microtesla, the time that it takes for alarm or precession is about 750 nanoseconds. So if birds are using this mechanism to sense the magnetic field of the Earth, it means that electron spin superpositions have to be alive for at least 750 nanoseconds, which is admittedly a tall order. It's not completely unheard of. For example, the nitrogen vacancy centers in diamond at room temperature live as a quantum sensor for about 2 microseconds. Okay? But, I mean, there's a reason why physicists like low temperatures and vacuum conditions, right? So this is a tall order, but it's not completely unheard of. So, which brings me to like my big existential and experimental problem, which is the fact that to date, there is no experiment on Earth that can unambiguously prove or refute whether superpositions can live for that long inside cells, for example, for a bird to sense the magnetic field of the Earth. So, in fact, if I needed to give a true title to my talk, it would be this. If superpositions are maintained for long enough inside cells, how can we learn to deterministically tweak them in a very particular way for function? Because in the end, birds fly and they orient, right? And even though the hypothesis was outrageous, experimental biophysicists liked the idea and started looking for animal, because birds, just like animal, because birds, animal proteins that could sustain electron spin-dependent chemical reactions. And the first animal protein that was known to sustain electron spin-dependent chemical reaction was this protein there called cryptochrome. And there's a question there. Yes, please.
Can you remind us what's the Zema energy at this field compared to the thermal energy?
Yes, so I'm going to preface this by saying that KBT is not an argument. I'm going to, again, preface this by using the example of the nitrogen vacancy center in diamond. So the qubit in the nitrogen vacancy center is about 3 gigahertz. That qubit is immersed into a, if you calculate KBT at room temperature in gigahertz units, of about 6,000 gigahertz. Well, and then you can say, doesn't the qubit thermalize? Well, it does, but it only yields quantum-enhanced information. It can only be used as a quantum sensor before thermalization. So things thermalize, but things are only interesting from the quantum point of view before thermalization. Here, I'm going to show you some energy, an energy diagram right now. The important part is actually not the Zeeman energy, is the hyperfine interaction, and which are very, very tiny. Okay, so KBT is not an issue here in the sense that it only works before superpositions die. Okay, it only works before thermalization kicks in. After that, there is no quantum sensing anymore. After that, magnetic fields do not alter the fate of chemical reactions anymore. And I'm going to show you exactly, I'm going to show you energy scales like 10 slides downstream. Bear with me, but that's the exact right sort of question to be asking, right? So back to cryptochrome. Cryptochrome is the first animal-found molecule to be able to sustain electron spin-dependent chemical reaction. It's present in the eyes of birds, in the antennas of migrating butterflies, but surprise, surprise, it's also present and expressed in all our human cells because it also has circadian rhythm regulation functions. Cryptochrome is a fluorescent protein. It has a flavin, a chromophore, okay? And in fact, the evidence that cryptochrome exists as a quantum sensor in nature is very widespread, but unfortunately at very disconnected length scales. So at the chemical length scales, there is no doubt that electron spins in cryptochromes are working as true quantum sensors, and here's how. There are 2 electrons that we care about. One electron comes from the flavin, the other electron actually comes from a nearby tryptophan. It's not super important. The way that we, in the lab, examining this protein is by putting it inside under a microscope, okay? And you shine laser light onto it. It doesn't need to be a laser, but it's more convenient if it's a laser. It's a laser in the blue. You get the flavin excited, and then it de-excites, emitting fluorescence, which you can then collect. Now, just like the spinning diamond, if you remember, cryptochrome also has a spin-dependent fluorescence intensity, which is a very convenient experimental handle. And it also gets its own exclamation mark because it's super cool. And here's why. And wait, because the energies are going to show up here. So imagine that you have 1 electron at the outer shell of the FAD, of the flavin. Okay? It's spin up, but you could have been spin down. It doesn't really matter. That's at the ground state. There's also an excited state that you can access by applying blue light. You shine light, electron gets excited, oops, de-excites by emitting light, which you then can collect. However, there's a competing pathway. Sometimes when you excite it, instead of it decaying down via fluorescence emission, it undergoes something that chemists call an intersystem crossing, whereby the electron sort of meets another electron, a free electron in this case, coming from the tryptophan. Now, both an external magnetic field and the internal hyperfine interaction is going to make the 2 spins, is going to put the 2 spins into a superposition of singlet and triplet. And again, the difference in energy between singlet and triplet is about 30 kilohertz. Okay, it's very, very tiny. So it's the hyperfine interaction that actually makes this superposition, and actually Zeeman interaction splits the triplets. I'm not showing it. This is a simplified picture. But at some point, the electron spin from the flavin wants to go down, to relax down to the ground state. It can do this while the two spins are in a singlet state, not emitting light via vibrationally assisted processes, or it can do so while the two spins are in a triplet. However, as given by the strengths of my arrows, it's way more likely for the flavin electron to go down if the spins are in a singlet state than if the spins are in a triplet state. So those two processes compete also with the fluorescing pathway. So, and here's the kick. And again, if you know the physics of nitrogen vacancy centers in diamond, I expect you to have memories of what I'm going to say here. Because on average, if you have one molecule undergoing this cycle many times, or many molecules undergoing this cycle once, on average, if your spin states are in the singlet state, because you have a larger chance to decay back to the pathway where you can emit light, on average, cryptochrome fluoresces a little bit more if you are in a singlet state than if you are in a triplet state. Okay, the amount of fluorescence difference is very, very tiny, but importantly, it's measurable. Okay, so by doing optically detected magnetic resonance, exactly like what you would do with a spinning diamond, you can actually do a true quantum state measurement of the electron spins. And again, if you have a microscope that is powerful enough to detect those differences in fluorescence energy, you have a true quantum microscope, a microscope that can tell you about the spin, like the true quantum states of two electron spins. And just to make sure, this is magnetosensitivity that doesn't involve iron at all. Okay, just to make sure it's all spin dependent. So there's no doubt that chemically things work out, and in fact people have probed the fluorescence of cryptochrome as a function of, in fact, of the time during which you applied a laser for cryptochromes in solution, so you are exciting this cycle many, many times. What you see is that the fluorescence is decaying, you're killing the chromophore, but as the researchers pulsed a tiny magnetic field on and off, you see a tiny modulation of this fluorescence intensity as a function of the magnetic field, as expected by the photophysics that I've just shown you. However, the next level of evidence comes from experiments with birds, with flies, with plates of cells that respond to magnetic fields in a way that is consistent with inside their cells there being electron spin-dependent chemical reactions, which, as I said, has never been unambiguously confirmed or refuted by no experiment on Earth. Can we do better? I think we can, which brings us to the final part of my talk, because this is not a talk about birds. This is not a talk about only cryptochromes. In fact, all of those cellular phenomena there have been shown to be tweakable by weak magnetic fields in a way that is consistent with this type of electron spin-dependent chemical reaction. And in fact, one very nice, a good indication that it might be spin-dependent is when you have an effect magnitude as a function of external field, not only going up, not only going down, but going up and down. And I can put a lot of papers here for you to see, but usually people get what I'm talking about when I show them this slide. This slide comes with a story. And actually, we just got some money. This is not my data, but we have just gotten some money to do a public replication of this experiment. I have a friend. His name is Peter Vierlinger. He is a bona fide precision measurement physicist. He builds the world's best hypomagnetic chambers, chambers that shield the magnetic field of the Earth or nearby magnetic fields very, very well. And usually what he does is he shoves ultra-cold atoms experiments inside. But he was starting to get requests from bio people to grow things inside his hypomagnetic chambers. And then he was like, okay, we're going to do this experiment. So here's what he did. He and his team of physicists grew tadpoles inside his hypomagnetic chambers that has a DC noise level at the nanotesla level, okay? Again, way tinier than the magnetic field of the Earth. And he grew tadpoles under two conditions. That he could only grow the tadpoles for 2 days, because that's as long as he could go before having to ask for a bio permit, because he's like a physicist. In the first run, he grew the tadpoles inside the chamber, but with a mimicked magnetic field of the Earth inside. And the tadpoles sort of looked normal. In another batch, he grew the tadpoles with only the residual 2 nanotesla field, and about 30 to 40% of the tadpoles were non-viable after 2 days. This has been reproduced by the group of Dima Budker in mice as well. And apparently, they're having trouble having the results published, because there is still a lot of stigma related to people talking about magnetic field effects in biology. So let's recap what I just told you. You actually don't even need to add a magnetic field. If you shield the tiny magnetic field of the Earth, the tadpoles really don't like. And nobody really knows what's going on. My best guess is that, so melanin, the stuff that gives the color to our skin, the stuff that gives the color to the tadpoles, there's very rich spin physics in melanin. If you ask me, I would, I think it has to do with melanin. And again, because the magnitudes of the fields involved are so tiny, everyone's best guess is that this is probably spin related. And again, quantum or not, there is no doubt that the tadpoles are sensing the magnetic field of the Earth, or the lack thereof. If they were doing this via magnetite, via a compass needle type of material, tadpoles would need to have big chunks of magnetite, which they're not known to possess. So everyone thinks that it is quantum related. And this opens up all sorts of questions like, what's the magnetic field on Mars? If you want to space farm in Mars, reproduce in Mars, can you do this? And answer, the magnetic field on Mars is 1 divided by 50 the magnetic field of the Earth. So that might have implications for space travel, who knows? So, and again, if the tadpoles are sensing the magnetic field of the Earth using a spin mechanism, it means that spin superpositions are available, are alive for at least 750 nanoseconds. However, coherence times requirements are way easier to achieve if you want things to sense, for example, the magnetic field of your cell phone speaker, which is on the order of a millisecond. For spins to sense the magnetic field of your cell phone using this mechanism, coherence times would only need to be as long as 50 nanoseconds, which is way shorter. And in fact, again, this is a hypothesis. I think this is what's going on some applications that people are actually using magnetic fields for. So people have found completely haphazardly magnetic fields that seem to be doing stuff to biology. For example, to the left, bottom left here, you see a picture from the website of a company called Novocure that is now valued at B with a B of ball, $billion. And it's a company that found a weak magnetic field that reduces the size of brain tumor. The company itself says they don't understand how it works. In orange there, it's what they say in their website. They say it's a combination of DNA damage yield, mitosis rates, they reduce the mitosis rates of cancer cells, reactive oxygen species modulation, and reorganization of cellular migration, cellular skeleton. And actually, those 4 things have been independently shown, some also by us, I'm going to show you our data in a second, to depend on weak magnetic fields in a way that is consistent with this type of electron spin-dependent chemical reaction. Now to the right, there are 3 pictures there, all having to do with not only one, not only 2, but 3 startups of Alfredo Obregon, a professor from the University of Singapore. He's like a biologist who knows his spin physics. Heuristically, he knows that cells that overexpress that TRPC1 gene are more susceptible to being magnetically tweaked. If anyone has any information about TRPC1 in this audience, please let me know, because I have no idea what this means. But he has 3 startups, one that reduces the size of breast cancer, the other one that does muscle regeneration, and the other one that increases the proliferation rate of lab-grown meat. So just by tweaking weak magnetic fields, he can do all of those 3 things. Again, all of this is super nice, except that it's a huge phase space of magnetic field, intensity, frequency, direction, and the model depends on all of those things. But this phase space, so far, for the past 40 years, have been explored haphazardly. I liken it to being in a haystack, grabbing a little bit of hay and finding a needle. So there is no predictive power. You cannot connect points in this phase space. You cannot connect 50 microtesla DC with 1 millitesla at 3 gigahertz, right? And again, you only get correlative data that might, or that seems to depend on spin physics. So I think it's time for us to break clean of that. So here's what I propose. I propose to get systematic causal data with predictive power by grabbing a microscope, taking a magnet, synchronizing everything down to the nanosecond, because that's the time scales of relevance for spin physics, and having very good photon detectors. Because, as I said, fluorescence intensity is a proxy, is a true quantum measurement of electron spin state. You put those 4 things together. Some people call it a glorified microscope. I call it a quantum instrument. And in fact, that's exactly the same type of instrument that is now used to study and control the spins in diamond. I want to do the same. I want to study and control the spins inside those proteins in the exact same way. If you do that, I argue that we can get causal data. If you add to that simulations, I'm going to show you one example, we get what I'm calling a code book on how to deterministically control spins in biology for function. You can actually, you might be able to write in your phase space in a deterministic way to tweak particular chemical reactions. So again, we have the scientific goal. I'm going to show you 2 optical microscopes that we're building. But our technological goal, which is completely science fiction now, I do really want to develop like electromagnetic interventions that I am most excited about, might provide some sort of therapy. Again, science fiction, but ideally I would love to have in my phone an app, and then you would click on an app and say, well, today I need help with wound healing. You click on wound healing in your app, and then your cell phone produces the exact magnetic field frequency and intensity that you need to do wound healing. Wound healing is something that people have been using magnetic fields for, again, super haphazardly. So let me tell you about the big experiments that are not online yet, but we've been building. That's the blueprint of all our microscopes. Again, 4 ingredients: microscope, the magnetic field, synchronization down to the nanosecond, and actually we use an open source software that is usually used by atomic and molecular physicists. And 4th, single photon resolution capabilities. We're building 2 microscopes. One is a lattice light sheet. It's being developed for the past 2 years. The second microscope is a turf that has been in development for the past almost 3 years. It's almost ready to take pictures, but not quite yet. So once those are online, here's what I think we can do with data taken from that. And that's how we can couple data from those instruments with simulation. Again, this is not only about cryptochromes, but cryptochrome is the poster child of this field. So there's historically, because of birds, there's plenty of data about cryptochrome. So you take the photophysics of cryptochrome that I showed you. Even though you cannot predict which molecule is going to be magnetic field sensitive, usually redox proteins, the proteins that are involved into the give and take of electrons, are usually related to magnetosensitivity. So you take the published redox cycle of cryptochrome, and you have enough parameters to set up a very simple open quantum system simulation. So for the experts in the talk, the minimal example, the minimal Hamiltonian that will give you this has 3 spins, 2 electron spins that start in a singlet state, but otherwise they don't need to have any interaction among themselves. So Zeeman of one electron, Zeeman of the other electron, and a third nuclear spin, which is coupled to only one electron, giving rise to a hyperfine interaction. With this model, you can predict everything that I told you about, again, this model predicts the dependency on magnetic field intensity, frequency, and for molecules that are attached to a membrane or with a certain orientation, there's also directionality dependence. And this is the type of result that you can get with this type of simulation. Have a look at this rightmost plot. In that rightmost plot, I plot the simulated, I'm dying to take this curve experimentally, the simulated magnetosensitivity of cryptochrome in solution, tumbling in solution, as a function of external magnetic field. So as you see, the magnetosensitivity as a function of external field, it goes up and down, as I said it would, good. Very cool. It peaks very close to the magnetic field of the Earth, which is either a coincidence or maybe it has to do with the fact that if birds are using that to follow the magnetic field of the Earth, maybe there has been some evolutionary selection for the protein to work better under the conditions where the birds are. We cannot tell those things apart. We think that people who can do directed evolution of proteins can tell us if you can actually influence proteins to be selected as a function of magnetic field. So again, with our big instruments, with this type of simulations, I think we can get a code book on how to drive those processes. But we're not there yet. Here's where we're at the short term. For the short term, we have been taking more correlative data on magnetic field effects in biology. Again, this type of data has existed for 40 years. They will not move the needle. The needle will be moved by the bigger instruments, but that's what we could do while we're waiting for our big instruments to be built. So we get our incubator, that's where cells live, and we can shove things inside. That's a commercially available hypermagnetic chamber, and we also developed an active set of temperature-controlled active coils, okay? And we can grow cells inside different magnetic field conditions. I would like to share with you some results. Most are reproducible. This is a null result, okay? Let me guide you through, and again, this is consistent with what's seen in the literature. So magnetic fields have been shown in the past to help wound healing. So here's what we did. We took muscle cells in a plate. Then what you do is scratch the cells. It's a scratch assay. There are controllable ways to do that. And then you monitor how well the scratch closes as a function of magnetic fields. Okay, and then you look at it, take pictures. And there you plot the area of the wound as a function of time of exposure of the magnetic field for different magnetic fields. And unfortunately, this is a null result. We do not see an effect on weak magnetic fields for wound healing. And then I was giving this talk to NIH. Guy raises their hand and say, when you do those experiments, are your cells happy? And like, I have no idea what you're talking about. And then he's like, did you put like serum media on, like, do you give your cells food while you're having them close the wound? And then we're like, yes, the cells are happy, they have media. And then he's like, try without the media, because apparently when the cells are unhappy, they do not have food, the pathway of wound healing is completely different. And actually, that apparently the second scenario mimics better what's happening in the body. So we're actually redoing this experiment without media to see if we have. Oui? Is it like the Amber Alert or something? Oh, shall I continue?
Yeah. Nothing's happening here.
Okay. Okay. Cool. Okay. So no result here. But here's a positive result. Pay attention, people. It's finally the positive result. Okay. Thank you. We can show in a way that I'm confident that is sort of reproducible that weak magnetic fields on the order of half your phones do seem to change cellular morphology. Okay, so your cells, as far as I understand, your cell's cytoskeleton is composed mainly of actin and microtubules, and weak magnetic fields can change the pattern of expression of actin, the green stuff in the first picture. They go from being long fibers to having like this weird shape. Microtubules, which are super important for cell division, for example, you can change the state of like how much they aggregate, polymerization rates of microtubules as a function of weak magnetic fields. In fact, if you disrupt microtubules, so microtubules are all over the cell, you can use a chemical called nocodazole to disrupt the microtubules, and then you actually look at this network reform. It turns out that for cells that are not exposed to magnetic fields, the microtubule network reforms, whereas for cells exposed to this weak magnetic field, for only 2 hours, the microtubule network does not seem to reform very well. So the ratio between tubulin and actin is very different depending on the weak magnetic field conditions. Also, we can mess up with the reactive oxygen species like cellular oxidants with weak magnetic fields. So cellular oxidants are important signaling molecules, but if you have too many of that, things go bad. So here's what we do. We get muscle cells and we either expose them to magnetic fields or not, right? And this is the ratio of the increase in production of H2O2, which is a cellular oxidant, as compared the exposed cells for the control cells as a function of time. So the one there is the no effect line. And we've seen, I die on the hill for that first point there because we've seen it once more. So for short exposure times, it seems that there is a 40% higher rate of production of this cellular oxidant for in the cells exposed to weak magnetic fields. Afterwards, things go down. We think there's some resilience process going on, but for those short times, we can apparently mess up with cellular oxidative states, which we also see in another assay where the fluorescence of a certain molecule is a proxy for the amount of reactive oxygen species. This is not yet reproduced, but there seems to be a difference in ROS, in reactive oxygen species amount, as a function of this magnetic field. Yes? The control is inside the magnetic field of the incubator. So here's what's crazy. Different incubators have slightly different magnetic field conditions. So our hypothesis is that there might be a lot of biological experiments that maybe don't reproduce, maybe they see slightly different magnetic fields, because in any case, it's a degree of freedom that people haven't been caring about. And I think that this needs to change, right? So I think that's more or less what I wanted to talk to you about. I made the run from the technological quantum sensor to maybe a living quantum sensor. I showed you a bunch of stuff, and I'll stop by saying, may the quantum be with you. What are your questions?
Yes.
The person for that is Margaret Ahmed from the Sorbonne in Paris. She's the expert on plant cryptochrome, and yes, she sees similar results. She can mess up with ROS production rates in plant cells in a way that is consistent with the stuff with weak magnetic fields. There's a whole lot of people concerned about, for example, can you increase crop production with weak magnetic fields, for example? Yes?
You might have mentioned this already, but is there a known spin-spin relaxation time for cryptochrome, like
Okay.
a T2 value?
So for cryptochrome in solution, T1 times are on the order of 1 microsecond at room temperature. Nobody has done this measurement, say, inside a living cell, which is exactly where we want to be, which is super important.
Very nice talk, and it's almost inspirational. And I understand the reluctancy of the life scientists thinking about it, sort of magnetic field. But biologists do value tremendously actual data. So it looks like right now it's still the quality of data that have yet to be improved, right? You mentioned about the tadpole abnormal percentage if you sort of remove the Earth's magnetic field. But biologists, value dose dependence, like what you have shown for the cells. If you show you have a point of 0 magnetic field, basically nano Tesla, and then if you have data for micro Tesla, right? And then the actual field is sub-milli Tesla, but then if you can easily do a few milli Tesla, so you have a whole range, you have a dose dependence. I think the data will be so convincing they cannot
I
look
agree,
the other
and
way.
that's why we're doing this reproduction, this experiment. And I completely agree. So the team in Mainz who did the second reproduction experiment from Peter Vierlinger, they have data showing that viability of embryos as a function of magnetic field, it increases until it gets to the magnetic field of the Earth. Because they were unaware of this quantum phenomena, they never went a little bit beyond the magnetic field of the Earth to see if it would continue to go up or went down. But there is at least one data set out there that is not published yet that shows this dose dependence. And I agree, we need more of this type of data. There was another question somewhere?
Actually, go back. Sorry, I'm kind of asking the same question. But the control, so is the natural, so we just don't apply any...
Control is magnetic field inside incubator.
Right, but then for the... so when you apply the external magnetic field, I'm wondering how you do that. Is it like a large coil? For the so when you apply an external magnetic field...
Yes, the external magnetic field is with that set of coils there.
Okay.
And it's water-cooled. Temperatures don't vary beyond 0.1 Celsius over days. Mm-hmm. So if the effects are temperature-driven, they have to be driven by delta T's smaller than 0.1 Celsius.
I'm sorry to interrupt, but I have this room reserved.
Oh, I'm sorry. Okay, sorry.
Well, let's thank our speaker and we'll continue.
No problem.